Dear viewers, Namaskar. Welcome to the lecture series on MOOCs in the subject Virology. Today's topic is Discovery, Nature, Origin and Evolutionary Importance of Viruses. The contents are as follows. First, Introduction. Second, Discovery of viruses. The third, Nature of viruses. Fourth, Origin of viruses. And at last, fifth, Evolutionary importance of viruses. To introduce the topic, it may come as a surprise to learn that every animal, plant and protist species present in the planet is infected with viruses. A virus is a communicable agent that can only replicate inside the organism's living cell. Viruses are obligate intracellular parasites. They occupy a no man's land between living and non-living world and have features of both. They differ from simplest microorganism, mainly bacteria, in several aspects. Impossible to observe under light microscope. They do not possess internal cellular structure. They possess only DNA or RNA, not both. They require appropriate living cell as host for replication. They are unable of metabolism. Individuals show no increase in size. When inside a host cell, viruses show some of the features of a living organism, such as the ability to replicate themselves, but outside the cell they are just inert chemical structures, thus fueling the debate as to whether they can be considered to be life forms. A particular virus has a limited host range, that is, it can infect only particular cell types. A virion is a whole viral particle having RNA or DNA enclosed by a protein shell, establishing infective form of a virus. Virions are not cells. They do not possess organelles except for the virions of the arenaviruses, which contain cell ribosomes that were packed when the virions were assembled. With all these facts, let us try to understand how viruses were discovered, nature of viruses, origin and evolutionary importance in this unit. Discovery of viruses. The invention of light microscopes helps it possible for visualizing the agents of bacterial, fungal and protozoan diseases. But the methods for visualizing and cultivating large microorganisms were virtually useless for viruses. The causative agent of smallpox and polioviral infection was not clear for many years, but it was known that the diseases were spread from person to person. The French bacteriologist Louis Pasteur was definitely on the correct path when he postulated that rabies was caused by a living thing smaller than bacteria, and in 1884 he could develop the first vaccine for rabies. Pasteur also anticipated the term virus, which is a Latin word, means poison, to specify this particular group of infectious agents. The germ theory of disease was proposed only about 100 years ago, that is at the end of 19th century, and pathologists came to a conclusion that causative organism will be there for each infectious diseases. Further, they believed that these agents of disease could be seen with the aid of a microscope, could be cultivated on a nutrient medium and could be retained by filters. There were admittedly a few organisms which were so fastidious that they could not be cultivated in vitro, literally in glass, meaning in the test tube, but the other two criteria were satisfied. However, A few years later, in 1892, Dmitry Ivanovsky was able to show that the causal agent of a mosaic disease of tobacco plants, manifesting as a discoloration of the leaf, filtered through a bacteria-proof filter and not able to seen or cultivated. Ivanovsky was unimpressed by his discovery, but Bizernik repeated the experiments in 1898 and became convinced this represented a novel form of infectious agent which he termed Contagium vivum fluidum, what we know as a virus. In the same year, Loeffler and Freud came to the same conclusion regarding the cause of foot and mouth disease. Furthermore, because foot and mouth disease could be passed from animal to animal with great dilution at each passage, the causative agent had to be reproducing and thus could not be a bacterial toxin. Viruses of other animals were soon discovered. Ellerman and Bang reported the cell-free transmission of chicken leukemia in 1908, and in 1911 Ross discovered that solid tumors of chickens could be transmitted by cell-free filtrate. These were the first indications that some viruses can cause cancer. Finally, bacterial viruses were discovered. In 1915, Chouart published an account of a glassy transformation of micrococci. He had been trying to culture the smallpox agent on agar plates, but the only growth obtained was of some contaminating micrococci. Upon prolonged incubation, some of the colonies took on a glassy appearance, and once this occurred, no bacteria could be subcultured from the affected colonies. If some of the glassy material was added to normal colonies, they too took on a similar appearance, even if the glassy material was first passed through very fine filters. Among the suggestions Chouart proposed to explain the phenomenon was the existence of a bacterial virus or the secretion by the bacteria of an enzyme which could lyse the producing cells. This idea of self-destruction by secreted enzymes was to prove controversial topic over the next decade. In 1917, D. Harrell observed a similar phenomenon in dysentery bacilli. He observed clear spots on lawns of such cells and resolved to find an explanation for them. Upon noting the lysis of broth cultures of pure dysentery bacilli by filtered emulsions of feces, he admittedly realized he was dealing with a bacterial virus. Since this virus was incapable of multiplying except at the expense of living bacteria, he called his virus a bacteriophage, literally bacterium eater or phage for short. Thus, the first definition of these new agents, the viruses, was presented entirely in negative terms. They could not be seen, could not be cultivated in the absence of cell, and most important of all, were not retained by bacteria-proof filters. Nature of viruses. Viruses are obligate intracellular parasite or subcellular infectious agents. They infect and utilize the host cell for its replication. The fully developed extracellular virus particle is called a virion. The virion have a genome that is either DNA or RNA enclosed in a protein coat known as capsid or nucleocapsid. Some viruses possesses a lipid envelope enclosing the nucleocapsid. They are enveloped. In that viruses, glycoproteins encoded by the virus are surrounded by the lipid envelope. The role of capsid or envelope is to defend the viral genome when it is in extracellular stage and to facilitate the entry of the genome into new susceptible cell. The structure of viruses will be explained in detail in our future unit. The nucleic acid genome of virus comprises the information required by the virus to replicate and develop new virions after its entry into a susceptible host. Virions bind to receptors on cell surface, and by methods explained below, the genome is released into the cytoplasm of the cell. Occasionally, the association with protein, that is uncoating. The genome redirects the cell's replication for the development of progeny virions. The cellular machinery which helps for the production of energy, that is ATP synthesis, and for the synthesis of macromolecule like translation of mRNA to produce proteins is essential. Proteins encoded in viral genomes usually belongs to three major classes. First, most viruses encode enzymes required for replication of the genome and the production of mRNA from it. RNA viruses must encode an RNA polymerase or replicase, since cells do not normally replicate RNA. Most DNA viruses have access to the cellular DNA replication machinery in the nucleus, but even so, many encode new DNA polymerases for the replication of their genomes. Even if they use cellular DNA polymerases, many DNA viruses encode at least an initiation protein for genome replication. An outline of the replication approaches used by different viruses is discussed below, and details of the replication strategy used by each viruses are discussed in future units. Second, viruses must encode proteins that helps in the assembly of progeny viruses. For simpler viruses, only one or a few structural proteins which accumulate with the genome to produce the progeny virion. Some complex viruses may encode scaffolding proteins needed for assembly which are not present in the virion. In certain cases, viral proteins essential for assembly may possess proteolytic activity. Detailed description of assembly of viruses are in our future units. Third, the larger viruses encode proteins which affect protection mechanisms of the host. These defenses include the immune response as well as the interferon response of vertebrates, which are highly advanced and effective methods for controlling or eradicating viral infection, and the DNA restriction system in bacteria, which are very useful in molecular biology and genetic engineering that control the introduction of foreign DNA. Vertebrate defenses against viruses and methods used by viruses against these defenses will be described in future unit. It is clear that viruses which have larger genomes can encode larger number of proteins. For example, herpes viruses in the family Herpes viridae have more complex life cycle and assembly than complex virions with small genomes. For example, poliovirus in the family Picorna viridae. The smallest non-defective viruses have genomes of about 3 kb, that is 1 kb is equal to 1000 nucleotides for single stranded genomes or 1000 base pairs for double stranded genomes. These small viruses may encode few proteins, for example 3 proteins in case of bacteriophage MS2. But for the largest known RNA viruses, the coronaviruses in the family Coronaviridae contain genomes larger than 30 kb, and the largest DNA viruses, pox viruses that belongs to the genera Entomofox virus A and C in the family Poxviridae contain genomes around 380 kb. These large DNA viruses can encode hundreds of proteins. It is the larger viruses that can afford the luxury of encoding proteins that interfere effectively with host defenses such as the immune system. It is meaningful recalling that even the largest viral genomes are small when compared to the size of the bacterial genome, that is around 2000 kb, and minute when compared to the size of the human genome. There are further subcellular infectious agents that are even smaller than viruses, which include satellite viruses, which depends on other viruses for its replication. Viroids, small, that is approximately 300 nucleotide, RNAs which are not translated but do not have capsid, and prions, that is communicable agents whose uniqueness remains controversial and it is made of only one protein. These agents are covered in our future units. Let us discuss now origin of viruses. The question of the origin of viruses is a fascinating topic, but as so often happens when hard evidence is scarce. Discussion can generate more heat than light. There are two popular theories. Viruses are either degenerate cells or vagrant genes. Just as fleas are descended from fleas by loss of wings, viruses may be derived from prokaryotic or eukaryotic cells that have dispensed with many of their cellular functions, that is degeneracy. Alternatively, some nucleic acid might have been transferred accidentally into the cell of different species, for example, through a wound or by sexual contact, and instead of being degraded as wound generally be the case, might have survived and replicated, that is escape. Although half a century has elapsed since these two theories were first proposed, we still do not have any firm indications if either or both are correct. Rapid sequencing of viral and cellular genomes is now providing data for computer analysis that is giving an ever better understanding of the relatedness of different viruses. However, while such analysis may identify the progenitors of a virus, they cannot decide between degeneracy and escape. It is doubtful that all presently known viruses have evolved from a single progenitor. Rather, viruses have perhaps arisen several times in the past by one or both of the mechanisms outlined above. Once formed, viruses are subject to evolutionary pressures just as are all other organisms, and this has led to the extraordinary diversity of viruses that exist today. Two processes that contribute significantly to virus evolution are recombination and mutation. Recombination takes place infrequently between the single molecule genomes of two related DNA or RNA viruses that are present in the same cell and generates a novel combination of genes. Of far greater significance is the potential for genetic exchange between related virus with segmented genomes. Here, whole functional genes are exchanged, and this type of recombination is called reassortment. The only restriction is the compatibility between the various individual segments making up the functional genome. Fortunately, this seems to be real barrier to the unlimited creation of new viruses, although it is not invincible. Since pandemic influenza A viruses can be created in this way. Mutation has certain significance in the evolution of RNA genomes, as in contrast to the DNA synthesis, there is no molecular proofreading mechanisms during RNA synthesis. Mutations accumulated at the rate of approximately 3 into 10 to the power of 4 per nucleotide per cycle of replication, whereas with the DNA this figure is 10 to the power of minus 9 to 10 to the power of minus 10 per nucleotide per cycle. In other words, an RNA virus can achieve in one generation the degree of genetic variation which would take an equivalent DNA genome between 3 lakhs and 30 lakhs generations to achieve. Once found by reassortment, an influenza virus evolves so rapidly that it takes only 4 years on an average to mutate sufficiently to escape recognition by host defenses to reinfect that same individual. And at last, we will discuss about evolutionary importance of viruses. 3 main mechanisms have been anticipated for the evolution of viruses. First one is escaped gene theory. Viruses originate from normal cellular nucleic acid and gain individuality from the cell. DNA viruses derived from plasmids are transposable elements, while RNA viruses derived from mRNA. The second one is regressive theory. Gradual disintegration of prokaryotes existing parasitically in eukaryotic cells, it is assumed that formation of enveloped forms like pox viruses are most probable through this way. The third one, co-evolution theory. Independent evolution in conjunction with cellular forms from primordial soup. Some scientists commented that it is improbable for the same mechanism to account for the diversity observed in viruses and therefore suggested that viruses must have progressed many times over. A study reproduced in 2004 contrary-wise proposes that all viruses share a common ancestor and may have originated before cellular life forms. In any discussion of evolution, it is pertinent to ask where viruses first came from. The non-appearance of fossil records or shortage of any evidences related to it have not prevented scientists from venturing about their origins. The 2 main opinions are that viruses have either originated. The first one is from degenerate cells which does not have the ability for independent life and are from fugitive fragments of cellular nucleic acid. The history of viruses extends as far back as man's historical written or pictorial records. The earliest references related to it was 3500 years old bas relief monument from Egypt revealing a man supporting himself with a crutch. Careful scrutiny knows that calf muscle of his right leg is weakened in the characteristic aftershock of paralytic poliomyelitis. Somewhat later excavation of the similar civilization found the withered body of the Pharaoh Ramesses V, whose face bears distinguished pock spots of smallpox. Such indications represents extensive association of these viruses with mankind. Overall, viruses are evolutionarily unstable and recombination, reassortment and mutation of viral genomes provide a platform for natural selection. Viruses have probably originated independently many times. It is likely that every living organism on this planet is infected